talk about metabolic pathways, and in particular, we are going to talk about glycolysis, pyruvate processing, the Krebs cycle, and the electron transport chain, which are collectively known as cellular respiration. I started with this slide because sometimes people get confused about what those four different things are, and they think that they're separate. Those four things that I just mentioned, again, glycolysis, pyruvate processing, the Krebs cycle, also known as the TCA, and the electron transport chain, are all part of cellular respiration. They are all connected in very important ways in that the products from each become the reactants for the next set of reactions. So let's look at an overview. These are the four processes that I just mentioned. You should know the stages and in what order they occur. And you should also know the major inputs and products, which include those molecules that move between each of the processes. You should also know where ATP is used and where it is made, because that is the goal of the entire pathway of cellular respiration. So cellular respiration is a key type of metabolic pathway that is used for making ATP, and it does this by starting out with glucose and slowly breaking it down. In these four different stages of cellular respiration, ATP or GTP, kind of is more or less the same molecule, at least for our purposes today, are made through two processes, and those two processes are glycolysis and the Krebs cycle, in glycolysis and the Krebs cycle. One of the processes is substrate level phosphorylation, which is when a phosphate group actually gets added to the glucose, or formerly glucose carbon skeleton, and then that phosphate group gets added onto ADP, which produces ATP. And the second process is through the action of ATP synthase, which is an enzyme which adds a phosphate group onto ADP to make ATP. And we see that only in the electron transport chain, although we see it in the electron transport chain in both cellular respiration and in photosynthesis. So let's move on to glycolysis. Glycolysis is a form of carbohydrate catabolism. What is catabolism? Well, recall that catabolic reactions are those that break down molecules, while anabolic reactions are those that build molecules or pathways. Now, in this whole long process of cellular respiration, we are slowly breaking down glucose. So we start out with glucose. That is our very first starting reactant. And we see glucose here. It could be linear, as it's represented here kind of abstractly, or it could be in a ring structure, as we see here. It's the same molecule, it has all the same atoms, either way that it's depicted in your textbook or diagram online or whatever you're looking at. So in the whole process of glycolysis, we have two different phases. The first phase is energy investment, and the second phase is energy harvest, or the payoff phase. Naturally, when we are investing energy, that means we are putting energy into the system. So in this series of reactions, and each arrow here represents an individual reaction or one step in this pathway. In this whole phase, we are using ATP and releasing ADP, and so we are investing energy into this process. Now, why do we do that? Well, we're not going to go into the detailed chemistry of it, but you could think of it as investing in a stock and waiting for it to pay off. Or in this case, we are using ATP to make a molecule that is going to yield more ATP than we originally put in. Because again, the whole point of this process is to get ATP. Now you'll see in the energy harvest phase, we get out 2 ATP for each piece of glucose. So actually what happens in the split arrow here is that we split glucose in half. And it's literally, we split it in half. We have 3 carbons in one half, 3 carbons in the other half. That's what is represented here. And we get 2 ATP made through substrate level phosphorylation for each of these halves of glucose. So total, from 1 molecule of glucose that we put in, we get out 4 ATP just from glycolysis. We invested 2 ATP, so we get out 4, subtract the 2 that you put in, so we still get a surplus of 2 compared to what we started with, right? The other things that we make in glycolysis are NADH, and that is just adding the hydrogen onto the molecule NAD+, and we do that twice again. And we also make pyruvic acid, also known as pyruvate, and this is actually the remainder of the carbon skeleton of glucose. So you see glucose here is represented by these 6 circles. We break it into 2 so that we have 3 circles on the top and 3 circles on the bottom. We've added a phosphate group as well from that ATP. And we're not going to talk too much about these steps in detail, but what comes out of glycolysis is pyruvate or pyruvic acid, and we have 2 of those because we literally broke the glucose molecule in half. Okay, so the next thing that happens is pyruvate processing. And pyruvate processing occurs inside the mitochondrion. So what is the mitochondrial job? Come on, say it with me. It's the powerhouse of the cell. I love when students say that because everybody just kind of giggles afterwards. So pyruvate processing is where we take the pyruvate, which is shown here, and again we have 2 of them. This is also known as pyruvic acid, as we said on the other slide. We pass it through the mitochondrial membrane, and then we convert it into acetyl-CoA. You'll notice here that pyruvate has 3 carbons, and acetyl-CoA only has 2 carbons. And don't count this as a carbon because this is an abbreviation. This CoA represents a much larger molecule that is often not drawn out because it's just so large it takes up a lot of space on the page essentially. I know it's kind of silly, but that does not count as a carbon from glucose. We only have 2 carbons from glucose left. Where did the other one go? It went into CO2. And so that is where all of the carbons from glucose are going to end up in CO2 molecules. And we see that we produce 2 CO2 molecules and 2 acetyl-CoAs. Because again, we have those 2 pieces of the carbon skeleton from glucose originally. Next, we go into the citric acid cycle, also known as the Krebs cycle, also known as the TCA or tricarboxylic acid cycle. Now, why do we have these different names? I know it's super annoying, right? The Krebs cycle name comes from the person who researched it first and published on it. The citric acid cycle name comes because after you put in acetyl-CoA, the next substrate is called citrate. So in other words, acetyl-CoA is converted into citrate. And then the tricarboxylic acid cycle, again, is a reference to that citrate here. So we start out with 2 acetyl-CoAs, and again, these are each 2 carbon molecules. And as those go in, they are combined with another molecule, which we'll talk about in a moment. There are different chemical processes that are occurring at each of these steps, which you do not have to know. What you have to know is that through this cycle, we're going to lose more carbons as carbon dioxide here and here. And when we are losing carbons as carbon dioxide, we're also producing NADH here and here. And you should know that we are producing GTP, and if you refer to that as ATP for my class, it's perfectly fine because it's a very similar molecule. And we also produce FADH2, which is similar to NADH, and we produce another NADH here. Great, so we produce a lot of NADH and FADH2, which is used in essentially the same way, which we've produced in the two other steps. I don't think I showed it on the pyruvate processing slide, but we produce it there as well. And in glycolysis, we produce GTP, which is essentially the same as ATP for our purposes, and that's also produced in glycolysis. And what else do we have from each of these different steps in cellular respiration? Well, we have the carbon skeleton, and that is what goes around here. So we are adding different chemical groups and moving them around, and we see that we lose one carbon here, another carbon here, but we still have 4 carbons left from the molecule. So we started out with 2, and then we combine our acetyl-CoA carbons with another molecule, as I said in the beginning, and I said I would get back to this. When we combine it with that molecule, we end up having 6 carbons. Now, a moment ago, on the last slide, we said that we were only counting carbons that came from glucose. Well, guess what? The 4 carbons that combined with the 2 carbons here also came from glucose, but a different glucose molecule. So that's why we could continue passing around this carbon skeleton, because we have carbon skeleton A from glucose A. We have carbon skeleton B, which combines with A to make the 6 carbon thing. And then we pass around the 6 carbon thing, make it into a 5 carbon thing, make it into a 4 carbon thing, and then we end up back with a 4 carbon thing. And that thing is called oxaloacetate, which you also don't need to know that name, but that is just the name of this molecule that has 4 carbons in it, and which is eventually combined with this 2 carbons of acetyl-CoA, or from acetyl-CoA. And that's where glucose ends up. Eventually, every single one of those carbons is going to be released as carbon dioxide. However, this is where the carbons stay. They never go on to the next step, which is the electron transport chain. Why do we need the electron transport chain? Well, how many ATPs have we produced so far? We produced 4 from glycolysis, but we also used 2, so we have a net of 2, and then we produce 1 in each cycle of the TCA, and we have 2 cycles per glucose molecule because we have 2 of those acetyl-CoAs, and so we produce 2 GTP. So that's not really that much ATP so far. We are expecting to make about 30 or 32 ATP per glucose molecule under really good conditions. So how do we get to that point? Well, the rest of the ATP are made in the electron transport chain. I know this says 24 ATP. There are lots of different calculations that you could do as a chemist to calculate how many ATP exactly are going to be produced, and it all depends on the exact concentration of all the molecules in the cell, and it's kind of hard to predict that. So we could say anywhere from 20 to 30. You could look it up online, and there are arguments amongst chemists about this number. Regardless, you should know that there's a good amount of ATP made per glucose, and those, the handful, the 5 or so that are made from glycolysis and the Krebs cycle, or actually I guess that's 6 total, that's not even near the total or the maximum that we can produce per glucose molecule. So what is the electron transport chain, and how does it connect with these other processes? Well, the electron transport chain is a series of proteins that are going to pass hydrogens or protons across a membrane to drive the passing of energy, which is eventually going to fuel this little tiny molecular machine called ATP synthase. Where do we get these protons? Well, they are donated by NADH and FADH2, which come from the Krebs cycle and also glycolysis and pyruvate processing. So let's take a look at the electron transport chain here. So this is a miniature representation of it, and we can see here that molecules such as NADH will donate a proton, becoming then NAD+, and the proton is pumped through the membrane. That is, it is moved across the membrane through a protein against its concentration gradient. That's important. So the protein here is actually using some of the energy from the proton, from the electron here, which is donated from the hydrogen, because remember, hydrogen is going to be one proton, one electron, and it's using that energy in order to pump that proton across. And then we're going to see that that electron is passed to the next protein, and so forth. So that electron is passed between all the proteins in the electron transport chain. They all get a little bit of energy from this electron. So think back to chemistry, or think back to the beginning of this course. We talked about the different energies of electrons and how electrons can be boosted in energy if they absorb energy. So for example, in photosynthesis, an electron is going to be boosted by light energy. So this is an energized electron, and it's slowly releasing that energy to these proteins. And they are using that energy to do two things, to do one or two things. The first one here is both passing the electron to the next protein, and it is pumping a proton. The second one here, represented by Q, it is only passing the electron, it's not pumping a proton. The third one here is going to both pass and pump. So you see there. And then this fourth one, cytochrome C, is going to just pass. So you see there. And then the final electron, our enzyme complex here, number 3, is going to take that energy from the electron, and it is going to use it to add the electron to oxygen, making water. And that is where the electron ends up. Okay, cool, right? We're all done. Oh wait, we didn't make any ATP yet. All right, so the final step in the electron transport chain is ATP synthase. So imagine that we've gone through this whole process a bunch of times. What is the result? Well, we're going to get a bunch of protons on one side of the membrane. So recall that molecules and electrons and protons as well tend to move from higher concentrations to lower concentrations, right? Well, we're keeping the protons from moving to a lower concentration across the membrane because they can't move across the membrane. Why is that? Because they are charged. Charged molecules cannot move across the membrane. So we're storing up all these positive charges on one side of the membrane. What is going to happen is that we do allow them to cross, but they have to cross by going through this enzyme called ATP synthase. So this looks kind of funky. It is literally a miniature machine, and what I mean by that is that it has these moving parts that interact based on chemical reactions. So we see all of the protons up here, and they're going to move through one at a time. They're going to spin this part right here as they go through, and that is going to fuel this other part to make ATP from ADP and an inorganic phosphate. So let's look at that in action. Again, we have all of the protons on one side of the membrane. We see that they are interacting with this part of the enzyme, which is rooted in the membrane. And these little dots, these yellowish dots, are actually those hydrogens. They take a little ride on this like carousel thing, and then they're spit out on the other side. And again, that's a favorable reaction because we're bringing them to the side of lower concentration. We're bringing them along their concentration gradient, so this is a very favorable reaction. And that is what allows this thing to turn, and then it's going to add a phosphate group to ADP. You don't have to know exactly how that happens. If you're interested in the chemistry, it is pretty neat. It literally kind of squishes the ADP and the phosphate together to the point where it's more favorable to just form a bond rather than be so close and not have a bond. But again, you don't need to know that. All you need to know is that ATP synthase allows protons across the membrane down their concentration gradient, and that movement literally fuels movement in the enzyme, which then leads to ATP production. It's a crank kind of situation. So the parts of this are called the stator. So this is the stator part, which is stationary. And then there's the rotor, which is the part which actually moves around. And you can see that here. I know it's upside down, but I made it upside down so that it would match this GIF. And so this is where we get so many ATP being made per glucose. The only thing that's really important here is that the protons stay in high concentration on this side of the membrane. If they don't, then the ATP synthase actually works in reverse, and it will use ATP to move to move protons back across the membrane to restore that gradient, because if that gradient is not in place, then it messes up the whole system. And remember, there are lots of these in the mitochondrial membrane. And so this one here might be trying to prevent the lack of protons that's adjacent to it over here. So I think that I've included all the links to these different GIFs, but also feel free to email me with questions. That summarizes cellular respiration, and I hope you enjoy.